What happens to time and biological clocks in the deep sea? The deep sea is blind to surface cycle, and it has long been seen as an arrhythmic black box. It's also very challenging to explore. We previously worked in the middle of the Atlantic using a submersible to dive to hydrothermal vents located at 1,700 meter depth. And there, a key species is the mussel Bathymoduolus azoricus. Tides influence abiotic factor at vents, so that the mussel environments alternates between the influence of the vent fluid, which is acidic and metal-rich, and oxygenated seawater. We established a protocol to fix mussel directly on the seafloor and freeze their time. And we looked at gene expression in the gills. And what we saw is that at vents, the dominant cycle is tidal. We have 2 yellow lines per 25 hours. We also have an about daily pattern. We have 1 yellow line per 25 hours in gene expression. We also expose mussel in the lab to light-dark cycle, and there we see that we mostly have a diel pattern of expression with a weaker but persisting also circotidal component. So the presence of a weak circadian pattern in the deep sea where there is no known daily cycle and of a persisting tidal component in the lab where there was no known, we didn't give any environmental tidal cycle, suggested that these rhythms could be driven by one or more endogenous clock, which we then wanted to assess. As it's very challenging to work with deep sea mussels, we first established primary cell cultures of Azoricus food cells, and after 3 weeks, we sampled them over time to determine if they possess endogenous clock. We worked with very limited amount of material, so we captured about 5,000 genes. Still, we could identify 157 circadian transcript and 509 circotidal one. The circadian transcript and stats are more robust, and they support the existence of a circadian clock. The circadian clock in other species is described as a feedback loop. So some activator can promote the expression of their target genes, whose resulting protein will eventually inhibit their own activators. We identified the potential clock genes in Azoricus, and we showed that these activators could indeed promote gene expression. We then looked at the potential repressor, and there actually period, which is present in both mammals and flies, is not acting as a main repressor in Azoricus. Instead, it's cryptochrome that is doing that. And period can modulate the repression activity of cryptochrome. So we hypothesized that in deep sea mussel, the circadian clock can switch between a diel or a tidal rhythm based on the per-criter ratio. Full details, data, and hopefully very nice discussion at the poster.
Number?
20.
Thank you. Okay, thank you, Audrey. So